Let's start off by defining the physical properties of a sound wave. We have here a sound source. Let's play this music again. So, the sound source generates sound waves. They propagate across the air, thereby generating areas of high and low pressures. So, it is the vibration of the air molecules that generates regions of high and low pressures. The high pressures are indicated by these dark circles, the amplitude, whereas the low pressure regions are the areas, the bright areas in between. So here we see the vibration of air molecules indicated by black dots. This is the source of this beautiful animation. We see here that the air molecules are vibrating, are moving in a longitudinal fashion across the air. When we say we have regions of high and low pressures, we mean the pressure rises or decreases relative to the air pressure, which is the equilibrium line here. The sound wave can be expressed mathematically as a sine wave. So, it has a wavelength, an amplitude, and a frequency. As we increase the amplitude, we increase the loudness of the sound. As we increase the frequency, we increase the pitch of the sound. Now let's take a look at the ear structure. We have here the external ear, the middle ear, and the inner ear. In the external ear, we have the pinna, which picks up the sound waves. It funnels the sound waves, and they are then transmitted through the ear canal to the middle ear. So the outer ear contains the pinna and the ear canal. It's filled with air. Then we have the middle ear. We have the tympanic membrane, or the eardrum, connected to malleus, incus, and stapes. These are three tiny bones, also called ossicles. The first bone, the malleus, is directly connected to the eardrum. So the incoming sound wave has a given frequency, and it causes the tympanic membrane to oscillate at the same frequency as the sound wave, moving inward and outward. And so do also these ossicles. Now, the function of these ossicles is to amplify the sound wave, which is of critical physiological importance, as we will see later on. The middle ear is also filled with air, like the outer ear. Finally, we have the inner ear. This is where we have the so-called cochlea. The cochlea is filled with fluid. Here I have a short summary. The cochlea is about 3 centimeters long, is a tapered tube containing 3 fluid-filled chambers that run the length of the tube. Let's take a look at the cochlea chambers. We have 3 chambers: scala vestibuli, scala media, and scala tympani. In all of the chambers, we have fluid. The fluid in the scala vestibuli and the scala tympani is the same. It's also known as perilymph. Now, the fluid in the scala media is called endolymph. We know that an extracellular fluid always has a high concentration of sodium and a low concentration of potassium, with one exception, namely of the fluid of the scala media. There we have a high concentration of potassium. This again is of crucial physiological importance for hearing, as we will see later on. We also have in the scala media the so-called organ of Corti. This is where we have the sensory cells. We have 2 types of cells in the ear: inner cells and outer cells. In this part, I will focus only on the inner cells. In a follow-up part, I will discuss the roles of the outer cells, which are actually the amplifiers of sound. the inner cells. They pick up the sound waves, they get deflected, so they get mechanically deflected, and this mechanical deflection is translated into electrical activity. Action potentials in the neurons connected to the inner hair cells, and these neurons are part of the cochlear nerve. The cochlear nerve collects the information, the action potentials, and takes this information all the way to the cortical cortex, to the auditory cortex, which I will also discuss in a separate part. The outer cells, they amplify the activity of the inner cells. But again, I will discuss the role of the outer cells in a separate part. Here we see in this beautiful image 3 rows of outer cells and 1 row of inner cells. Humans generally have about 12,000 outer hair cells and about 3,500 inner hair cells. Unfortunately, if they come to harm, they cannot regenerate. We can also see that these cells are supported by a so-called basilar membrane, and I will discuss the role of this basilar membrane later on. Now, to the function of the ear. Here I have the summary. We have the outer ear, again the pinna, and the external canal. So the outer ear collects and funnels sounds. The middle ear, where we have the eardrum, malleus, incus, and stapes, the ossicles, the middle ear amplifies sounds. Then we have the inner ear. It decodes sounds. So the inner hair cells, they translate the mechanical activation into electrical signals that are picked up by the cochlear nerve as action potentials transmitted to the auditory cortex. Here's the summary. The function of the ear. Essentially, the function of the ears is to receive in the outer ear, amplify in the middle ear, and convert sound in the inner ear into a neural impulse, an electrical signal that is sent to the auditory cortex, as we will see in a separate part. So here we have the inner ear, the cochlear. There we have the inner cells. They get deflected. I will come back to this later on. As they get deflected, they get excited. Eventually, the neurons connected to the inner cells, they generate action potentials that are collected here in the cochlear nerve, which goes all the way to the auditory cortex. For this, we need several neurons, and I will discuss these neurons in a separate part. Let's take a look at the path of the sound wave in the ear. We mentioned earlier that both the exterior and the middle ear are filled with air. It's much easier for the sound wave to move across air, and it's much harder for the sound wave to be moved across a much denser medium, such as the inner ear, which is filled with liquid. So, when the sound wave arrives at the middle ear, it will have to be transmitted to the inner ear against a much higher resistance, the much denser fluid. So how does it do so? Well, we have to compensate for the loss of energy of the sound wave, or else we won't be able to hear, or barely. So this loss of energy that will be compensated for is referred to as impedance matching. There are several steps. Before we delve into the details, here again, the sound wave arrives at the middle ear, it oscillates the eardrum, then the ossicles, and eventually, here we have the oval window. This is the entrance into the inner ear, and then we have to move this sound wave through a medium which is much denser than air. So we have the impedance matching, which is required, or else we would barely be able to hear the sound wave. There are 2 principles. The first is physical, and the second also. Basically, pressure is force over area. P is F over A. So we can increase the pressure by increasing the force or decreasing the area. Let's take the first example. decreasing the area of the stapes. Remember, the stapes is here connected to the oval window, the interest to the inner ear. We see here the eardrum and here the stapes, that is the last ossicle in the middle ear. The ratio, the surface ratio between the eardrum and the stapes is about 14 to 1 or 20 to 1. Now, this information may vary in different textbooks. It is somewhere between 14 to 20-fold, the ratio of the areas, which means that the area in the stapes is reduced about 14 to 20-fold. So we reduce the area 14 to 20-fold, thereby increase the pressure 14 to 20-fold. Again, based on the same principle, pressure is force over area, we have here the ability to increase the force. We increase the force, we increase the pressure. Here we need the so-called principle of lever. So, like the seesaw, we have a long arm of the lever and a short arm of the lever. By this principle, we can here increase the force significantly at the oval window, again at the interest to the inner ear. We can increase the force greatly as compared to the force generated here at the malleus. To simplify this, we can consider the two bones, the malleus and the incus, as one, whereas stapes is considered as the second. So, the entity of malleus and incus can be considered as one lever, the longer arm of the lever, and the stapes would be the shorter arm. Here I have the summary. The first ossicle, the malleus, remember this one here, has a long arm that is attached to the inside of the eardrum. Here, the malleus is attached to the eardrum. The malleus is then linked to a second ossicle, the incus. This is the incus, the second ossicle, at an angle so that together they form a folded type one lever. So this is the principle in physics called folded type one lever. The malleus and the incus form a type 1 lever, the longer arm of the lever. An inward movement of the eardrum causes the folded lever to rotate counterclockwise, and the internal arm of the incus forces the foot plate, which is here the stapes, to press in on the oval window. You see here, this is the long arm, the malleus and the incus, the type 1 lever, and here's the shorter arm, the stapes. You can see the example here again. So we can here increase the force significantly. This is the second principle of impedance matching. So now we have the summary of principle 1 and 2 of impedance matching. The ratio of effective eardrum to oval window size is roughly constant at an average of 19 to 1. Remember, the textbooks vary between 14 to 20. And in this source, they say 19. The mechanical advantage created by the middle ear bones is also relatively constant, with an average around 2.4 to 1. Some sources say 1.4 to 1. Now to the basilar membrane. The basilar membrane, remember, in the scala media and the organ of Corti, it supports the hair cells, supports the function of the Corti organ. Now, the basilar membrane has physical properties that vary depending on the position along the basilar membrane. The beginning of the basilar membrane is known as the base. At the end, we have the apex. And the physical properties vary strongly. This enables the basilar membrane to separate the frequencies of the sound waves, as we will see later on. We also see that the basilar membrane, which is here, supports the inner and outer cells. Again, in this part, I will only deal with inner hair cells, and in a separate part, we will discuss the roles of the outer hair cells, the amplifiers of sound. As the sound wave reaches the inner ear, it also reaches the basilar membrane and causes displacement. So the basilar membrane is displaced. The sound wave has a given frequency. And as it reaches the basilar membrane, it generates a so-called traveling wave or standing wave. It has certain characteristics which we will discuss in the following slides. Now we see here in this beautiful animation how the basilar membrane is displaced by the incoming sound wave. Now this sound wave has a certain frequency and it reaches the maximum of displacement in the basilar membrane, which is specific to the frequency of the sound wave. We will see in the next slides. Here we will discuss the characteristics of the traveling wave. This is a sound wave that causes displacement of the basilar membrane. This displacement is known as the traveling wave. Let's take a look at the characteristics of the traveling wave. Regardless of the frequency of the wave, the shape is almost identical. So it starts here very low, and the amplitude builds up and reaches the maximum at the specific region of the basilar membrane that codes for the frequency of the sound wave. Assume we have a sound wave that has a frequency of 10 kilohertz. The sound wave starts here very low, the amplitude builds up, and eventually it reaches here the peak. If the sound wave has a frequency of 10 kilohertz, the peak of this traveling wave will cause this amplitude at a point in the basilar membrane that also codes for 10 kilohertz frequency. If we had a frequency of the sound wave of 5 kilohertz, this peak will be reached at the point of 5 kilohertz. This means that the basilar membrane distinguishes between different frequencies of sound waves. We have a dynamic range between 20 kilohertz in the very beginning at the base and 10 hertz at the very end, at the apex. And this is due to different physical properties of the basilar membrane, among other things, as we will see in the next slide. So, the separation of the sound frequencies along the basilar membrane is known as tonotopic representation. The determinants are, first, physical properties, and second, neural encoding, as we will see in the following slides. But we will start off with the physical properties of the basilar membrane. Here we have the base, and here we have the apex. The base is narrower and thicker and stiffer than the apex. We see here the apex gets wider, thinner, and also softer. So here we have the stiffest region, and here we have the softest region. It is the variation of the physical properties along the basilar membrane that enables the basilar membrane to separate the frequencies of the sound, thereby generating the dynamic range human ears, which is at the base about 20 kilohertz. This is the highest frequency, so the sound waves with the highest energy, with the highest frequency in human ears, about 20 kilohertz, they cause the displacement of the basilar membrane, the strongest displacement at the base. Along the cochlea, the frequency decreases. So, the sound waves with the highest frequency, with the highest energy, arrive at the base, and the sound waves with the lowest frequency, with the lowest energy, at the apex. This is the tonotopic representation, physical properties of the basilar membrane. We see here in this beautiful animation how the basilar membrane is deflected. Here too. Now to the second principle of tonotopic representation, neural encoding. Assume we have a sound wave with 1.6 kilohertz. So it causes the traveling wave, which peaks at a point in the basilar membrane, which is connected to a neuron that also codes for 1.6 kilohertz. If the traveling wave reaches the peak at 800 hertz, we have here a neuron in the basilar membrane that codes for the frequency of 800 hertz, and so on and so forth for the other frequencies as we see here. So this is the second principle, neural encoding. Now these neurons form the cochlear nerve. They take this information all the way to the auditory cortex, where we have the same frequency coding. I will discuss this also in a separate part. Finally, the mechanotransduction. This is the process where the deflection of the inner cells in the ear, so the mechanical deflection, is converted into electrical signals that generate in the neurons electrical signals, so action potentials, that are carried away by the cochlear nerve to the auditory cortex. So, for the process of mechanotransduction, we need to know the following. The endocochlear potential. We have here the endolymph. Remember, the endolymph here has a high concentration of potassium, okay? Now, the potential between the endolymph here and the inner hair cell is about minus 70 millivolts. So this is the potential difference between the endolymph and the inner hair cell. Potassium concentration in the endolymph is very high. It is slightly higher than the concentration of potassium inside the inner hair cell. So there's a slight chemical force driving potassium ions into the inner cells. However, there's a much higher electrical force driving potassium into the cell. So we have a force driving potassium ions into the inner cells. The question is, how do potassium ions go into the cell? They go into the cell through these channels, which we see here. These are mechanosensitive potassium channels. The sound wave deflects these hairs. These hairs are known as stereocilia, the shorter ones, and the longest one is generally called the kinocilium. So the deflection of these hairs toward the kinocilium pull on the hair cells. Now, the hair cells, as we can see here, are connected to one another by structures that are known as tipplings, which we will also see in the next slide. So, the mechanical wave, the sound wave, deflects these stereocilia toward the kinocilium. This stretches the tipplings, and the stretched tipplings pull on the mechanosensitive potassium channels, as we see here. These potassium channels then open up. Since we have a driving force for potassium to go into the cell, potassium will go into the cell. Now we have here calcium channels, voltage-gated calcium channels. Potassium, which polarizes the inner cell, opens up the calcium channels, calcium flows into the inner cell, activates the vesicles to release the neurotransmitter glutamate. Now, glutamate binds to receptors in the neurons, which results in action potentials, as we see here. Deflection of the inner cells, as we can see here, generates a higher frequency of action potentials. I would like to stress, we always have some activity going on, as we can see here by this frequency of action potentials. Now, this frequency of action potentials can increase as we deflect these cells. The action potentials that are generated in the neurons upon the deflection of the hair cells, okay, these action potentials that are generated in neurons are collected by the cochlear nerve and carried all the way. Remember, we need several neurons, but I will discuss this in a separate part. So these electrical signals as action potentials are taken by the cochlear nerve all the way to the auditory cortex.